Okay, well, thank you very much for the introduction. So I was asked somewhat at the last minute to be the first speaker as opposed to speaking later in the week because Jim Sethian wasn't able to be here. Of course, the organizers may live to regret this because while I am going to talk about something that's free and something that has a boundary, I'm not sure that it conforms to the Ockendonian definition of a free boundary problem. So I'm prepared to take various arrows and tomatoes from the third row there if I don't quite fit into the natural classification of things. Okay, so I'm going to tell you today about a project that is very much in progress. It's a combination of experimental work and theoretical work, and it has to do with a problem in developmental biology. Basically, a beautiful organism like you see here, which is an alga about 0.5 millimeter across called Volvox that has daughter colonies on the inside that grow up, and at some point in their life cycle they do an amazing thing. They turn themselves inside out. And that's the problem that we want to try to understand. But I thought it was important to, especially being the first speaker and in a conference of this type, to put this whole question in context. In other words, why on earth are we worrying about this, and why should you worry about problems of this type? So the context is really problems in evolutionary biology. And if I asked you to make a list of the really big problems in biology, and by this I don't mean will the funding go up next year, I mean things like, the origin of life. That's surely the biggest unsolved problem in biology. And probably the next one is the nature of consciousness. But right below that, I think many people would agree, the question of the origins of multicellularity is a very big unsolved problem. That is, historically, evolutionarily, how was it and why was it that the simplest unicellular organisms that first appeared on Earth 1 billion years ago or more eventually evolved to become larger and more complex? Larger meaning having more cells, but still not necessarily dividing up those functions of life into different cell types, but eventually becoming like we are, full of many different kinds of cells that have specialized behaviors. It's a very simply stated question, and there are many groups in many kinds of academic disciplines all around the world worrying about this question. So that's what I'm going to sort of have sit over much of what I'm talking about today. So first of all, the bulk of the work I'll be describing has been done in collaboration with Stephanie Hone and Aurelia Honnerkamp-Smith, who are two postdocs of mine right back there. Raise your hands. Right? Stephanie's a biologist, Aurelia is a chemist by training, and Pierre Haas, who's an applied mathematician, a graduate student in my group, and various other members of the group I'll mention as we go on. So I've passed around 3D glasses because at some point I will show a 3D movie, so you can look forward to that. To put this all in context about the evolution of, let's call it, biological complexity, in which we talk about the development of many different cell types, I think it's useful to show this plot. And some of you might have seen this before if you've heard me speak on other problems in biology. But I think it's one of the most interesting plots that one can show in evolutionary biology and basically biology in general. So it's a log-log plot of the number of distinct types of cells found in the body of organisms whose total cell number is down here, classified according to the different types of living organisms, green algae, animals, fungi, etc. So this was first presented to the world by Bell and Moores, and it was sort of represented and popularized by John Bonner, a very distinguished evolutionary biologist at Princeton who's about 92 by now, I think. So what you see here is that there's a general trend as we go from small organisms like, say, a bacterium that would live at 1,1 and obviously does all the functions of life with a single cell, to larger organisms like a human who would have something like 10 to the 14th cells. After all, we're about 1/10 of a cubic meter, and a cell is about 10 microns on the side, so that's 10 to the 14th cells. And humans, by this measure of counting, have about 210 distinct cell types. So if there were a bunch of statistical physicists in this audience, they might say, I see a straight line through all this data, and there must be a power law, and let's worry about that exponent. But I don't think that's very useful, because A, the data is incredibly noisy, and then how are we going to talk about all these different organisms in such a simple way? Maybe there's a way, but I don't think that's the case. But you do see that there's a general trend that as things get larger, they get more complex. So if we're going to try to make some progress and understand how this happens, I submit, and many others have submitted over the past 100 years or so, that the place to begin is the lower left-hand corner here, where we ask ourselves, as we look at a set of related organisms that, say, have ever-increasing cell number from 1 onward, there may be a point at which they jump and have 2 cell types, or 3. And I think that if we can't understand why that transition happens, we're probably not going to understand much more of this large graph. Now, in biology, when these big questions arise, it's generally the case that one looks for model organisms to study. So if you're interested in genetics, of course, it was the fruit fly. If you're interested in the wiring of the brain, you look at C. elegans, the nematode, where you can count all the neurons and all sorts of things like that. So the discovery of the right model organism for these kinds of problems was reported recently by a man named Antony van Leeuwenhoek. You all know the name? Raise your hand if you don't know the name. Oh, okay. He was the perfecter of the microscope, not the inventor, but the perfecter, who figured out how to make amazingly good microscopes with resolution far beyond what was there previously in the late 1600s. And he wrote many letters to the Royal Society, and this is my favorite. It's what he found in the animalcula he found in the excrements of frogs and other such things. He was looking at basically pond water, and you could do this yourself if you go to the River Cam and scoop it up. You'll find what he found, many green organisms that take the form of a spherical structure with many green dots on the surface and smaller spheres on the inside, which he studied and found quite fascinating for many years. Now, this organism we now know as Volvox because it was named as such by Linnaeus, who named everything originally in biology, for its persistent rolling action, the Latin root volvere, which means to roll, or the car, Volvo, same kind of root. This rolling motion is a consequence of flagella that live on the surface of this organism, and in a coordinated fashion allow it to swim in a very beautiful way. Now, I brought van Leeuwenhoek's microscope. Well, I brought a copy of van Leeuwenhoek's microscope, which I picked up in the Boerhaave Museum in Leiden. And here he is, the hero of today's talk. And this microscope is amazing. It has a single lens, about 1 millimeter or 2 across. It has a little translation stage. It's all of 5 centimeters large, and he discovered everything. He discovered bacteria, biofilms, you name it. He figured it out using just this little thing. So please pass it around. Whoops. And have a look. Okay, so Volvox is a photosynthetic, multicellular green algae. It's typically on the order of 0.5 millimeter to 1 millimeter across. And ever since the late 1800s, it has been recognized as a model for many of these problems in evolutionary biology, and we'll now see also for developmental biology. And it was August Weismann who was perhaps the most outspoken proponent of this point of view. And it was echoed by David Kirk in a classic book on Volvox in the late 1990s, that there are basically 7 or 8 main reasons why we should find this a model organism. First of all, it and many of its cousins, which range from unicellular to vastly multicellular, live now. We don't have to look in the fossil record and figure out what was going on. We can grow them. They're readily obtainable in nature. As I say, you can scoop up water from the River Cam and you'll find them. They're really easy to obtain, easy to study from many different perspectives, genetic, biochemical, developmental. So we have a kind of holistic view of how they operate. And we know something about their ecological niches, so we know what's driving where they live. Also, their appearance on Earth and differentiation is recent enough that genomes of both the single and multicellular organisms can be related in a confident way to understand what changes led to multicellularity. And there are interesting evidences for repeated genetic changes leading to this form, and it's amenable to modern molecular biology methods, although not so easy. Okay, the other thing to say is that this organism is spherical. And for those of us who have at least half of our brain doing theory, that's really important, because everything is much more symmetric and easy to think about. So that's the spherical green cow. So here's a family portrait of these organisms to give you an idea of what one can see just by scooping up pond water. So the unicellular member of what is known as the Volvocene green algae is called Chlamydomonas, clammy to its friends. It's about 10 microns across. You can't see here, but it has 2 flagella that allow it to swim with a breaststroke through its fluid environment. And then there is a whole set of species, this is just a subset, that have 2 to the n, where n runs up to about 15 or 16. Chlamydomonas-like cells that are arranged in a kind of clump for small n, and by the time you get to larger n, they exist as a spherical form with a transparent extracellular matrix that holds them together, a kind of rigid gelatinous structure, and they're mounted, in this case, on the outside with their flagella pointing out. And this continues all the way up to Volvox gigas, which can be something like 3 or 4 millimeters across and have 50,000 cells on it. So it spans a huge range of sizes, from 10 microns to a few millimeters, and a huge range of cell numbers. Now, what's very interesting here is that obviously this organism, having only one cell, does all the functions of life with one cell. But by the time you get to the larger species, you discover that there's a dividing line such that everybody to the left continues to have cells that do all the functions of life, and for that I mean specifically reproduction and everything else. Watching the BBC, everything else is lumped together, but reproduction is special. But at this point, there appear 2 cell types. Sterile somatic cells that are on the surface and have the flagella pointing out. They are like the galley slaves that just keep swimming. And specialized reproductive cells that live on the interior and eventually grow up to be the next generation. So this is what is known as germ, for reproduction, soma, somatic, sterile, differentiation. And it's the most elementary differentiation in biology that you can think of between cell types. So, of course, there are many interesting questions, like why did nature put this dividing line here? What's so special about this number of cells or this size that makes it evolutionarily advantageous to be like this? That's a separate question, but it's a big one. And I think this is the place where we can look to try to answer questions about complexity in a precise way. But today we're going to focus in particular on the development of these daughter colonies and how they evolve. But I want to just point out that the fact that nature's presented us with this whole spectrum of sizes and behaviors actually makes these organisms models for biological fluid dynamics. I've just written a big review article that points out all the things you can do with it. It's like one of these multi-purpose tools you can see advertised on television. You can study multicellularity. You can study interesting hydrodynamic phenomena in which these organisms orbit each other and remain attracted to each other purely by means of a proximity to a no-slip surface. You can use them to measure very precisely the flow fields created by thousands of interacting flagella and test hydrodynamic theories of how you connect flagellar action at the microscale to the large-scale flow. You can interrogate individual cells like Chlamydomonas and the beating of their flagella with great precision and try to understand how they synchronize with each other. That's one of the great questions in this area is the nature of synchronization. You can ask yourself a question, how, if I don't have a central nervous system, but I have 1,000 independent parts, do I manage to steer toward the light when the light shines from some direction I'm not pointing? And that's called phototaxis. And you can also look at some interesting fluid dynamical problems involving tracer statistics and suspension. So there are many, many things that one can work on here. Okay, but let's just talk about Volvox. So here is a nice picture of Volvox sitting on a micropipette, the same kind that you might use for in vitro fertilization or something like that. It's about 3 or 4 or 500 microns across. You see the individual flagella on the surface, and of course the only way it can swim is if the flagella point out. That's sort of an obvious statement, but let me just say that. So this is the typical way that it swims. This is using a tracking microscope that we built about 100 meters from here, in which the organism is swimming up through a light sheet up against gravity. Basically, the daughter colonies that you see here, the larger, the intermediate-sized white blobs, cluster in the posterior, so the organism's bottom heavy. So like a ship with a heavy keel, it rights itself and swims up. And we have a camera that's on a microscope stage that's moving up with it by feedback control. So we're in its frame of reference. You can see these microspheres around here. You see this beautiful turning motion that comes from the fact that the flagella point from the North Pole to the South Pole with a slight east-west twist. So it spins as it swims. And you can actually do particle image velocimetry, a method using the tracing of microspheres to construct the flow field as a function of distance from this organism and test those theories I mentioned before. So that's the normal way that the adult behaves. And in fact, if you look in detail at the, let's say, the anterior North Pole of the colony, you'll see that every cell has a little red dot, which is an eye spot, a primitive photosensor that allows this whole organism to do phototaxis. And if this is a measure of where the North Pole really is, you'll notice that there's a great degree of order in this pattern. They all point down and out away from the North Pole. And if you take a colony and smush it against a glass slide and put a little blue arrow pointing from the center of each cell to the eye spot, what you see looks a lot like what a condensed matter physicist would think is a magnetic spin collection, right? It's not perfectly ordered, but it's pretty well ordered. And that's an example of what's called planar cell polarity. So this just shows you that there's a lot of structure here. It's not a random collection of 1,000 cells. It's highly ordered, highly structured, and reproducible. And one of the questions in developmental biology is, how do you get that?
Is there a light source that's polarizing this? What's giving the bias?
Ah, well, in fact, that was what I was just trying to say. How on earth does it make this happen without random processes disrupting it? I won't answer that, but maybe in the questions I can talk a little about it. Okay, so let's talk about the life cycle of this organism, and then I'll finally get to the point of today's talk. So Volvox carteri, which is the workhorse organism that we use, has a very fast and exciting 48-hour life cycle. And it works something like this. Let's pick it up here where we have an adult which is giving birth, let's say, to daughter colonies. So we'll start that at the beginning. We'll call that like the first day of Genesis. So when these daughter colonies are formed, they're small, maybe 100, 150 microns across, and they have inside of them a small number of 6, 8, 10 germ cells. These are single cells, undifferentiated, undivided, which during the first day, and this is a day-night cycle of light and dark, during the first day, the organism grows. These germ cells grow very large without division, and then at the end of that, they grow through repeated rounds of cell division, something like 10 divisions or more, to produce the 2 to the 10 cells necessary for the daughter colonies to be like the mother. However, when they're first produced, those next generation cells, colonies, have their germ cells, on the outside, and their flagella pointing inward. And that won't work if you're going to try to swim, right? So there's an amazing process called inversion. And here are some static pictures, electron micrographs. Whoops, what happened there? Static pictures, electron micrographs of this process in which, for instance, in Volvox carteri, a cross-shaped hole opens up at the top of this little sphere. 4 lips peel back, and it turns itself inside out. That takes tens of minutes, an hour, or something like that. And then these guys are now in the right orientation. And then the rest of the life cycle is just a kind of inflation, like a balloon being blown up. The total cell number is fixed. All that's happening is everybody's moving further and further apart. So this makes for a very interesting life cycle, a very interesting epoch right here where you can study many fluid dynamical issues as a function of size. But today we're going to focus on this process of inversion. Now this turning oneself inside out reminds you perhaps of a process known as gastrulation, which is an early embryonic phenomenon in which an embryonic mass of cells starts invaginating to produce the gastric system. Now, the famous biologist Lewis Wolpert said this: it is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life, because if it doesn't happen, you're not here. So there are some very interesting similarities between these two processes. And in fact, there's a whole field of evolutionary developmental biology known as evo-devo, which basically compares developmental processes of different organisms to understand the evolution of those processes. And of course, you're probably all familiar with this now mostly discredited idea of ontogeny recapitulates phylogeny. That is, that as an embryo is developing, it's kind of going through the historical sequence of all the things that happened evolutionarily beforehand. It's not completely wrong in the sense that there are structures and phenomena that you see, for instance, in the development of animal embryos in which you have this invagination and producing gastrulation, and this may have been the kind of thing which, for a primitive colony of multicellular form, led to the first digestive cavity that might have existed. So there's the possible connection between these two to keep in mind as we think about this process. Now, let's talk a little bit about the details of this inversion process in Volvox. So remember, there are many different Volvox species, many different sizes, and there are 2 main types of inversion, type A and type B. Here is a movie showing you Volvox carteri, which is the type A inversion, which is the one I mentioned before, where there's 4 lips peel off, and the whole thing then turns itself inside out. So basically, the wave of shape transformation begins at the top and progresses down. Type B inversion, which you see here for Volvox globator, is quite different. There is an invagination that's produced at the equator that constricts and then produces this overturning effect that eventually leads to the inversion process. And then there's a sort of intermediate type, which for one or two species is known, where maybe both things are happening. But it's a free boundary problem, right? I mean, nobody's guiding this. There are no hands making this happen. It just happens. And we're trying to understand how this occurs mechanically, dynamically, and obviously biologically. So the basic underlying idea that has been put forth over the course of many years, and it's a pictorial idea, is that there is a wave of cell shape changes that occurs here and that is driving everything. So let's look at what this would imply for type B inversion. So the first thing I have to tell you is that all the cells that are in the pre-inverted state and in the post-inverted state are connected together by little nanometric structures called cytoplasmic bridges. So these are stretchable, elastic connections between cells, and there are multiple connections tying everybody together. The extracellular matrix, which is what holds everybody together in the adult and holds them far apart, doesn't yet exist. So all we have are these cytoplasmic bridges, and you could think of this red line as the locus of all of those bridges between neighboring cells. Now, the cells at the equator in the type B inversion are known to elongate into structures known as flask-shaped cells. So they're long and skinny. And moreover, that the cytoplasmic bridges that connect them, which used to connect them pretty much at the midline, actually migrate down to the end. So it's quite plausible that if I have a bunch of, let's call them ellipsoidal shaped objects, and I connect them together at the base, they're going to splay. And so that creates a region in which the local curvature would like to be as shown here, rather than convex outward. And that this continues to happen, pulling this region in. There can be cell shape changes also at the top, but this wave of progressive cell shape changes just drives sequentially the whole pattern to turn itself inside out. So in a sense, you could think of it as driven by the combination of cell shape changes and migration of cytoplasmic bridges. And then by the end of the process, the cells go back to the shape that they had before, and everybody's happy, and it's just maybe a slightly different shape, but not flask-like. And it's happy and inverted. Okay, now, we know from various studies that these cytoplasmic bridges move. Electron micrographs can show where the cytoplasmic bridges are, but more importantly, as in many times in biology, various mutations give us a key to what's happening. And there was a particular mutation studied in this work involving a gene called INV-A that codes for a motor protein called kinesin, which walks along microtubules, which are filamentary structures inside the cell. And those are responsible for translating the location of the cytoplasmic bridges, or the cell relative to the bridge, and making that splay happen. And in mutants lacking that, the inversion process stops halfway through. So basically, the cells can't continue this process of shape change and then reversion back to the original structure. And so the picture is basically that there's a body of microtubules inside the cell, and there are molecular motors which are basically walking along those and translating the cell so that you produce this splay. And if that doesn't happen, or it happens only partially, then you can't actually develop the proper inversion. Okay, now the interesting thing I find about this is that because we have this entire spectrum of cell sizes from Chlamydomonas all the way up to Volvox, there actually is a whole sequence of inversions going from essentially the flipping back and forth of a little plate-like structure for the organism called gonium, which has 16 cells and basically looks like a flat plate with a bit of curvature, all the way up to these very much more complicated structures where the invagination and cell shape changes are taking place in a wave. So the ultimate goal of all of this would be to try to develop a theory that can explain the sequence of shape changes that we have over size, but we're not there yet. Okay, so the classic way of trying to understand in any detail what's actually happening over the course of time is not to use time at all, but actually to stop the organism's development at some point and put it under the electron microscope, or even worse, embed it in some gelatinous material and make slices of it. And this has been done heroically by Stephanie and many others, and it's shown a huge amount of important insight into what's going on. So here are electron micrographs at various stages of the inversion process. And most importantly, here are these cross-sectional studies. So these are very thin sections of a few microns or less, and you can see exactly the cell shape changes that led to those pictures. It was, of course, these that led to the schematic diagram. So there's no doubt that this is happening. But what one would really like is some way to see this process where you could identify every cell in real time, take slices optically, and measure everything. And that's what's been missing. So it turns out over the last 5 or 10 years or so, there's been a wave of interest in a microscopy technique known as SPIM, selective plane illumination microscopy. And it's essentially a very simple technology, but it's become very popular by virtue of various open source initiatives. And the idea is basically to take a laser, create a light sheet from it using a cylindrical lens, send that through a microscope objective so that it's a very, very thin sheet, pass it through the sample. Here you see a little Volvox embedded in a gelatinous material like agarose, sitting on a support which can be rotated or translated so that you can have the light sheet slice in different positions. And then you look orthogonally to that at fluorescence that comes off. And so you have a light sheet this way, and you just see a cross-section of the organism as you step through. Now, something like this is what is known as confocal microscopy in the sense of producing sections. But that works on a very, very small scale, and we're trying to talk about organisms that are millimeters or several millimeters, and that's not possible with standard confocal microscopy. Now, actually, I know that probably very few of you are experimentalists, but just for you to appreciate how generous various people are with their time and effort. This is the webpage of OpenSPIM. You go there, and they give you a parts list. Basically, they say, buy this part from this company, buy this part from this company, little videos of how to put it together. You need to do some local specialist construction, and we have a fantastic machine shop here that has made this possible. And there you go. So it's like an open access article, but it's an open access experiment. And I think that that's a really commendable effort on this group's part. Okay, so here's what it looks like in reality. It's about this big. We pick a laser off from down below where we have several lasers available. We basically bring it through various lenses and produce the light sheet, and we send it through a microscope objective here. Here's the chamber, and here's our camera that picks up the fluorescence looking orthogonally. And basically there's a little motorized drive that allows us to turn the sample and to translate it. So it's really quite straightforward. Okay, so here's an example of what cross-sections look like as you take them. Every few microns, you're translating the sample. And of course, most of Volvox is empty on the inside, so occasionally the daughter colonies show up, and then you go to the other side and whoosh, there you go. But of course, that's not really how you want to watch it. What you want to do is put your 3D glasses on. Now, we've never done this before, right? So I hope this works. Aurelia and Stephanie. Oh, I wish I had a camera to take a picture of all this. This is fantastic.
I'm about to do that.
You're about to do oh, fantastic. So let's see how this looks. It's a little tricky to get. Maybe the lights have to go further down. Can you see it? It takes. You have to relax your eyes. Just let it happen. Yeah. So what you're seeing here is over the course of many tens of minutes, individual colonies or daughter colonies are deciding to invert. Typical inversion process is 30 minutes, 40 minutes, something like this. And so they're not perfectly synchronized, but the whole colony is also, the mother colony is also growing during this time. So it's a very interesting set of problems here about how is this extracellular matrix added to allow the cells to move away from each other so homogeneously, but more importantly, how is this inversion process happening? Okay, so I'll show you another one. So this one is a close-up of one of those embryos. And the thing to notice is you can see its germ cells on the outside, and then whoosh, now they're on the inside. Right, so that was a little too fast, perhaps, but it's very clear that it's oriented the wrong way, and that it wraps around and goes like this.
Is that real time?
No. It takes tens of minutes, yeah. Okay, but now the next one is really interesting. I think this is one of the best movies we have. This is Volvox globator. So this is type B inversion, where it invaginates along the equator. And then you'll see this thing comes up, whoosh, and this little mouth goes closed. Right?
What is rewarding that behavior?
What is?
What is rewarding that behavior? Why do they do that?
They do this because there's an existential question here. Why on earth did nature do it this way? But when the embryos are first produced, they're inside out. So there has to be a way to fix that. So this is to fix that. Now, we can talk about why on earth biology preserved this error that they then have to, then goes have to fix, right? I don't know, but that's what it does. Okay. Enjoy that? Good. You can take them off now. No, Stephanie and Aurelia, this is a heroic feat. So there are others, you know, we call this four-dimensional imaging in the sense that it's space and time. And as you see this going up, of course, you're not restricted to this view with modern digital technology. You can take a slice in any direction and start measuring things. And with help from my PhD student, Philip Cooktrong, I can show you just a taste of what's possible using MATLAB to basically go through these images and quantify things. So you can extract I hope that's going to run, please. Oh, come on. Here it goes. Did it run? This is always tricky to do. Yes, it's running slowly. I don't know why. Well, good. I can talk over it. So this is a particular cross-section of type A inversion, and you can see the cross-sectional shape. You can see the development of these lips folding back. You can measure all sorts of things. You can measure the curvature. You can construct tangent vectors. You can actually measure the arc length, at least as well as you can estimate it, to try to understand the competition between bending and stretching. I mean, the cells change shape. There's certainly a fair amount of that that's going on, but there's also the possibility of real stretching. So from the point of view of mechanics, this is a problem with bending and stretching and intrinsic curvature, and that's what we're trying to put together here. So this is the beginnings of a quantification of all of this that's only made possible by this three-dimensional imaging where you can slice. Okay, so let me get out of that, back to here. So now I just want to spend the last few minutes talking about how to build some kind of theory for this. And let me just say at the start that I think the way to proceed, which we don't often do in mathematics, is to test a hypothesis. I mean, of course, in biology, that's what you must do. You have to say, I'm testing a hypothesis. We don't often work that way. But I think here we have to. So the hypothesis that has been advanced is that a wave of cell shape changes is sufficient, let's say, to induce this phenomenon. Is there something else needed? We don't know. So let's see where we can go with a sequence of cell shape changes. Now, when I first learned about this, I was reminded of something in the world of membrane physics. So in the world of membrane physics, when one talks about things like the shape of a red blood cell or lipid vesicles or things of that type, it's natural to wheel out what's known as the Coulomb-Helfrich elastic energy. So this is a quadratic form in the two objects that you construct for a surface, obviously, the mean curvature, the sum of the inverse principal curvatures, and the Gaussian curvature. And it looks something like this. It's an integral over the surface of the mean curvature possibly minus some constant squared plus the Gaussian curvature. So that's the general quadratic form that you'd write out. Now, the integral of the Gaussian curvature over a closed surface, of course, is a topological invariant, so on some level we don't care about that. But the thing that's often hypothesized in membrane physics is that there is this spontaneous curvature, this preferred offset for the curvature. And it was called the spontaneous curvature. In those cases, it may play a role even in the shape of the red blood cell. You might say, well, this shape, closed bag that has these indentations, comes about by just deflating a sphere, and it has to put dimples somewhere, and it ends up there. But actually, if you wash away the membrane, which you can do with detergent, you end up with a network of what's called spectrin, which is a polymer that's anchored in the membrane, and it still has the shape. It's imprinted on it. So that's a case of intrinsic curvature. And in the membrane physics world, there are many situations where this can arise. So for instance, the lipids that make up membranes can be of different shapes on the outer or inner leaflet. There can be proteins that actually cluster on one side of the membrane and clamp it. That's responsible for certain kinds of endocytosis. There can be proteins that preferentially go only on one side of the membrane. There are many, many examples where you can get this kind of spontaneous curvature by an asymmetry between the two surfaces. But here it's really very simple. We know that there's this splay coming about from the cell shape changes. So the essence of this is essentially to do what you would do in the Helfrich theory, but to do it in the context of the elastic theory of thin shells. So the basic idea, which you can look up in standard references and has been discussed a lot recently with interest in buckling of spheres, is to, first of all, talk about a reference system, which you imagine is sort of in equilibrium, and then to refer the deformations of the system back to that reference system. So for instance, if you had an axisymmetric spherical shape as your reference system, well, you could talk about the radius as a function of arc length. Obviously, this angle is irrelevant. And then there's a tangent angle here, which you can just relate by geometry. And then the actual shape would have a different arc length parameterization, S, and the trick is to talk about the relationship between a point on the surface that's at arc length S that used to be at some arc length S naught. And that gives you an idea of the stretching that's occurred in the various directions. So for instance, if you look at these quantities involving this stretching, you can construct the strains in the meridional and azimuthal directions. And from this, you can construct an elastic energy as a generalized quadratic form. So it's going to involve the Young's modulus of the system, because that's the fundamental resistance to stretching, the Poisson ratio nu, and it will involve quadratic forms in these strains and also in what are known as bending strains that are related to the curvature of the actual system relative to the curvature of the reference system. And this is essentially, to leading order, the same as the Helfrich theory, but it's done in the context of the theory of elastic shells. So what we're going to do is just propose that there is a wave of bending of intrinsic curvature and see what that does to the equilibrium shape. So it's a static problem. There's no real dynamics here. We're basically saying at each point in time where we construct a region shown in red of preferred curvature, we will solve to find the equilibrium shape and then enlarge that over time and see what happens. And this calculation, which is hot off the press from Pierre, shows that you can get very far just by increasing, letting the system equilibrate, increasing the width of this preferred curvature and driving the system to something that looks an awful lot like what you see in type B inversion. Now, there is some discussion in the literature about whether some kind of active contraction has to take place on the bottom here to make this sort of snap-through happen. And so one of the questions we're going to try to answer is, can we compare these kinds of results with, say, the velocity of this motion or any other geometric quantities to decide whether that active component of compression is necessary? Yes?
Is the length constrained?
No, there's stretching allowed. Yeah. Okay, so this is just a measure. It's noisy here because this is a tricky numerics. This is just a measure of the actual energy of the system as this process is occurring. Of course, you're putting energy in by making these cell shape changes and making the spontaneous curvature change over time. But at least it shows that this basic idea that a wave of cell shape changes could be responsible for the observed pattern is not completely wrong. And I hope the next time I see you, we'll be able to have put this together with the experiments to see if this is really correct. Okay, so there are many, many questions that one could proceed from here. Fine if we say, yes, there are equatorial shape changes that make this kind of purse string that pulls things in and then pulls it up. But how did nature know to start it there? What's the signal that said start it at the equator and not somewhere else? So that's one of the key questions. Is there some active contraction in the posterior? And I think most importantly, the larger view here is we have this whole set of organisms of different sizes, and we know that there are systematics. Is it just a matter of size? Is this a matter of fitting in some mode into the system? Or how has nature decided to deal with these as a function of size? And secondly, the growth of the extracellular matrix. How is this all added afterwards in such a way as to preserve the local order of the cells on the surface that allows it to swim and do all of the functions that we know? Okay, thank you. I think I'll stop there and happy to take questions.
Thank you very much.
Strikes me as one sort of straightforward topological question that comes to mind is, in this Volvox quaternary, it seems that the cytoplasmic bridge disruption occurs first, whereas in the globator, the disruption of the cytoplasmic bridge occurs last. Now,
Well-
we have this, Gauss-Bonnet theorem that tells you the outer normals have to add up to a certain total. But that seems to have been disrupted initially in the quaternary form, where it tears open.
At the top, yes, and then,
Right.
exactly.
And then it can flip around and invert, whereas in that globator, you get this
Absolutely.
invagination, and the disruption
Absolutely.
is...
It seems to stretch,
very
and then,
last stage.
exactly. Yep.
Could you say a few words about that?
Well, I think that's exactly right. That's an incredibly important distinction. And in fact, early on, I was trying to understand why are there 4 lips when it disrupts at the top. Is it hardwired by just the geometry, or the what's the word, the valence, the number of cytoplasmic bridges connecting different cells? Is that what makes it break into 4? Or maybe it's actually just sort of pre-patterned by the division process that produced the whole organism. But for sure, there's some critical stress that is necessary, and then those bridges break. And I don't know the answer, but I agree that that's a very important thing, because it's clear there's a lot of stretching and orientational changes of the cells in type B at the top before it finally says, okay, I'll tear and go around.
Yeah, so nature sort of took 2 very different routes there. It's very
Yeah.
interesting.
It's fascinating, yeah, but we don't know the answer.
More questions? Oh, yeah.
How about the fluctuation? I mean, if I look at your experimental results, they seem to be quite consistent in terms of the time it starts to get inverse process, and they occur in certain locations, but in a system like this, quite far from equilibrium, you have fluctuations.
Well, in fact, I think this might be the right movie. You see, it's sort of sitting there going, I'm going to try. There's a lot of quivering that goes on. That's not thermal fluctuations. This is clearly just because it's too large, I think, for that to be the case. But this is some kind of coordination that's going on, and perhaps what happens first is lack of perfect coordination, and then it finally goes. So the question of how much communication is necessary from one cell to the next to make this happen, in particular if this wave of cell shape changes is to really work, then it has to be not suddenly nucleated over there. It has to progress along. So is this communicated through the cytoplasmic bridges? I mean, that's one of the things that cytoplasmic bridges allow, is cell-cell communication. That could be the answer, or it could be just geometric. There's some forces as a consequence of the shape changes that trigger the next one to go. And surely there's noise in that because it's a real biological system.
Ray, in your original descriptions, which were almost photographic, you emphasize the discrete nature of the elements making up the system and the interaction between discrete elements. But as soon as you introduced any mathematical structure to the system, you immediately went into a continuous. Is there a possibility that that's actually going to be a more difficult transition than is thought?
So I only plead prejudice in doing that. I think that there's a natural tendency to want to coarse grain these things and to think of it in terms of some continuous theory. But if we're going to understand issues like how does this wave of cell shape changes occur, obviously that's at the discrete level. And there are mathematical formulations of the gastrulation process. Actually, my student Jocelyn, who's somewhere back there, worked on for some time, in which you actually just make discrete elastic elements, you couple them together with springs and stretchable elements, and you can get the same thing to happen. So I can see both points of view. I think that this picture of preferred curvature is an appealing one because it's the one that comes out from the cross sections and the biological narrative. So let's see where we can go with that. But I readily admit that there would be things we can't handle with that. And also the validity of it may break down in terms of the size, the curvature relative to the thickness. At some point we're no longer going to be in that valid state, but we close our eyes and see.
I was taught, perhaps incorrectly, that that blood cell shape you showed is an optimization of the volume of the cell and the rate of diffusion through the cell membrane. That that particular shape optimizes, in some sense, those two factors. But you haven't mentioned any of the physical processes other than these proteins moving around. Could, again, we be looking at some sort of interaction of diffusion of either nutrients or something, which is also accounting for maybe the topological change?
So that's an interesting point about the red blood cell shape. I'm surprised by that explanation because at least the academic path I took wheeled out specific calculations where you look at elastic membrane deflated a certain amount, you look at red blood cell shapes, it's a perfect match between the two. And I would think that these diffusive processes would be much less sensitive to the detailed shape. I would think that they care about more gross features of the structure.
I think the idea, though, was you trade off volume for thinning the cell.
In that sense, yeah.
Get the more rapid
Well, that's probably a
through the
consequence.
membrane
Yeah.
of oxygen, I guess.
Yeah,
to the
okay,
red cell.
right, that makes sense. So I would see that as a consequence of the elastic driving force, but nature picked it. I mean, evolution was at work, so obviously it's not just a question of satisfying some theorist's ideal. It's a question of functioning in the body, and surely that would have happened. So I like that. I don't immediately see how to translate that here, but that's why at the moment I say, this isn't our theory and we're going to pin our hopes on it. This is us being able to try to test a hypothesis, and maybe we'll find that it doesn't work, that we can't explain various things, in which case we'll go back. Yeah.
Okay. More questions? Well, if not, then let's thank Ray for this beautiful talk.